Hey, Assalamu alaikum. Today I am talking about the topic of molecular diagnostics or genomics or genetics and molecular biology is fluorescence in situ hybridization. It is also called FISH. Okay. By its name, it tells us the whole story as it starts from the word fluorescence. Okay. By word fluorescence, it means it produces a color. Okay. By its name, it tells us it produces a somewhat color. And the in situ mean it is inside, it is inside the cell. Okay. By its name, it tells us it's a fluorescence or a color produces inside the cell. And the word hybridization means it produces it binds with somewhat else. Okay. For example, it is a template. It's like a template, and the fluorescence is attached with it is that. Okay, it's attached here, and by its name, it tells us it produces a fluorescence when it attaches with the DNA template or a chromosome. Okay, and by this, it produces a color. It produces a somewhat color. Okay. Okay, now we'll talk about the introduction of the fluorescence sensor to hybridization. As we call, the fish is a method widely used to detect. It detects the protein, RNA, as well as DNA structures in place in the cell. Okay, if it is a cell and DNA, protein, or RNA is present inside the cell, it detects it. It detects it without extracting outside. Okay, without extracting outside. uh, it is actually the importance of fish or of fluorescent in situ hybridization. That is why it is called in situ. Okay, fish. Fish targets specific sequences of chromosomes. Okay, with fluorescent probes. Okay, as we talk here, as we talk in the previous slide, it is a DNA. Okay, it is a DNA, and probe is attached here. Okay, and by this fluorescence is produced when it hybridizes with the actual uh, DNA tablet. Okay, now the word here is the fluorescent probes. Okay, what is probe? Probe is actually the short single-stranded sequence SSS. Okay, short short single-stranded sequence. Okay, that is complementary, that is complementary to the DNA or RNA or the protein. Okay, and when it binds with the DNA, RNA, or protein, it produces fluorescence. Okay, that is why it is called fluorescent in situ hybrid. Sorry, fluorescent probes. Now, probes are designed to hybridize to the critical areas that are amplified, deleted, translocated, or otherwise rearranged in the disease states. Okay. Now, actual thing is probes are designed. Okay. As we say in the bioinformatics, the primers are designed. The probes are also designed. Okay. How it designed? For example, this is the whole DNA. Okay. For example, it is the DNA, or we can say that it is the genome. Okay. Now, the problem or the you can say the mutation is present here. Okay, the mutation is present here. The rest of the template or the rest of the genome is all okay. So we designed the probes that attached from here to here. Okay, that attached just in this place. Okay, by this we can check it. We can detect it. Okay, by this we can detect it. The word is critical areas. Actually, the critical areas are the places or the area that is actually have a problem. Okay, that are actually the somewhat is amplified. Amplified means the nucleotide is actually added or inserted into the template, or the nucleotide or or the sequence or the base pairs are deleted. Okay, or the chromosome is translocated. Okay, for example, translocated is the what is translocation? Translocation, it is the exchange of the parts between the two non-homologous chromosomes. For example, it is the chromosome 9 and it is the chromosome 22, for example, and the part of the chromosome is exchanged between these two chromosomes. Okay, these two chromosomes are not homologous; they are non-homologous chromosomes. Okay, the parts are exchanged between that. It is called translocations, or otherwise rearranged in diseases, or maybe other type of diseases in which this type things are occur. Okay, and if amplification, deletion, or translocation occurs, we used we design probes to hybridize that critical areas. Okay. Now, fish analysis requires a fluorescence microscope. And now, the main thing is because we use the fluorescent probe. Okay. Now, the detection of the fluorescence can be done by using fluorescence microscope. Okay. Now, the detection of the fluorescence can be done by fluorescence microscopes. So fish analysis requires a fluorescence microscope. It will excite fluorescent emission for the probes. Okay. Uh, fluorescence is produced, fluorescence is attached. Okay, when we use the fluorescence microscope, it will excite. Okay, the fluorescent is excite for the probes. The actually, for example, it is a probe. Okay, it is a probe. Fluorescent dyes are attached here. Okay, when we analyze in the fluorescent, in when we analyze it in the fluorescence microscope, it excites this fluorescent dyes. Okay, it excites this fluorescent dyes. By this, these fluorescents are excited. Okay, these fluorescents are excited, and it produces a color. Okay, and when it produces a color, we detect it. Okay, and special filters for the detection of fluors emitting at different wavelengths. Okay, uh, it tells about the special filters. Actually, uh, for example, we have to analyze the 14 places, the 14 areas of the chromosome. Okay, we have to analyze the 14 areas of the chromosome, but we have just seven probes. Okay, we have just seven probes. So when so we have to increase the numbers of these probes in order to analyze 14 areas of the chromosome, or we can use seven special filters, special filters in order to analyze these 14 areas of the chromosome. For example, if we have, for example, if we have white color, okay, and we mix it with, with red color, it produces a pink color. Okay, it produces a pink color. So, so we use seven special filters, and by this we can analyze the more areas of the chromosomes. Now there are two types of the fish. Okay, one is interface fish, and second one is the metaphase fish. Now, as the uh, other types of the analysis of the chromosome or the gene is karyotyping and bending. Okay, karyotyping and bending. Now, the problem is for karyotyping, we have to grow the cell. Okay, we have to grow the cell, and at metaphase stage, we have to arrest the chromosome. Okay, we have to arrest the chromosome in order to analyze or detect the disease or the problems of the chromosomes. Okay, but in interphase phase, we don't need to culture the cells. Okay, we don't need to grow the cells because because growing cells in culture is not required. Why? Why? Because in interphase phase, we don't need to arrest the chromosomes or the cell at metaphase state. Okay, it can detect the chromosomes or the problems or the mutations or the deletions or the amplifications or the translocation at interphase stage. So we don't need to grow the cells. Interface-based methods are used commonly. Interface-based methods are used commonly to study prenatal samples. Okay, prenatal samples, tumors, and hematological malignancies. Hematological malignancies means blood problems. Why we can commonly studies these types prenatal samples of tumors and hematological malignancies because not all of which are conveniently brought into metaphysics in culture because these prenatal samples, tumors, and hematological malignancies are not conveniently brought into metaphysics in culture. They don't go through the metaphysics stage because they don't grow, and we can't restrict it at the metaphysics stage. Okay, so we use the interface phase to detect it. Now we come to the methods of the interface phase. Actually, for phage cytogenetic analysis, fixed cells are permeabilized and exposed to the probe. First of all, first of all, the is probe is permeabilized into the fixed cell. For example, it is a cell. Okay, it is a cell, and it is a nucleus in it, and this is the genetic material or the chromosomes. Now, the probe is permeabilized into the cell. Okay, the genetic material is not extracted out of the cell, but the probe is permeabilized and exposed. Okay, now the probe is permeabilized into the cell. Okay, now the probe is permeabilized into the cell, and by this analysis can occur. The probe is 60 to 200 kb fragment of a DNA. Okay, now it is the properties of the DNA or it is the actually the definition or the characteristics of the probe. It is 60 to 200 kb. Okay, the size of the probe is 60 to 200 kb fragment, and it is attached covalently to the fluorescent molecule. Okay, probe, it is a fragment and it is attached with the fluorescent dye. Okay, the fluorescent molecule covalently attached, and this fluorescent molecule is actually excited by fluorescent microscope, and when it is excited by fluorescent molecule, sorry, microscope, we detect. The probe will hybridize or bind to its complementary sequence in the cellular DNA. As we all know, the probe is actually complementary because single, it is a short single-stranded sequence. Okay, and it is complementary. Okay, it is specifically bind with the cellular sequence. Okay, so it is a specific and it is a complementary to the cellular DNA. Now, the bound probe is visualized under a fluorescent microscope, okay, as a point of fluorescent light. For example, it is a cell, it is a DNA, sorry, it is a nucleus, and it is microscope, oh sorry, chromosome. Now, probe is attached with the complementary sequence of the DNA that is present in the microscope. Okay. Now, when we saw it in the fluorescent microscope, we saw the points. Okay, we saw the points in the cell. Okay, we saw point of fluorescent light in the nucleus of the cell. Now probes are designed to complementary to the particular chromosome, okay, or a chromosome locus, so that the image under the microscope will correlate with the state of the chromosome or the locus. It means there are total 23 pairs of chromosomes. Okay, total 23 pairs of chromosomes. Now, one probe is complementary to the one pair of the chromosomes. Okay, one probe is complementary to the one pair of the chromosomes. One pair of the chromosomes. It means if it binds with the pair of the chromosomes, okay, if it binds with the pair of the chromosomes, the two two points will be 2 points of the fluorescent molecule will be seen under the microscope. As we see here, it is a normal cell in which there are 2 points. Okay, there are two fluorescent molecules or the points are shown here. Okay, the probes hybridized to the chromosomes. But if there is a triploidy, okay, if there is a triploidy means there are one extra chromosomes present in the cell. There will be three. There will be three fluorescence will be shown. And if there is a deletion, then there is just a 1 point. And the centrometric probe for a normal diploid cell, triploidy or trisomy, center and deletion of the monosomy, the right. Okay. Now it is the type of the probe that is dual color probe. In dual color probe, we use different types of the probes. Means one color is yellow. Okay. The color of the one probe is yellow, and the second one is green. Okay. So we use two types of the probes. It also has a have a applications we will talk uh, here. Actually, the multiple probes, if we use the multiple probes, okay, that is as we say, the one probe is complementary to the one pair of the chromosomes, and if we use the multiple chromosomes, sorry, multiple probes, it will bind to the more region of the chromosome. Okay, that is why here is multiple probes spanning large regions are used to detect regional deletions. Okay, if multiple probes that are spanning the large regions, regions means, for example, it is a chromosome. Okay, it is a chromosome. And one probe is attached here, 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 and one probe is attached here. By this, we can detect the deletions, the regional deletions. That regional deletions means the region of the chromosomes if deletion is occurred. For example, if deletion is occurred here, okay, the deletion is occurred here, and by this, these probes, these two probes are not attached here, okay, the two probes are not attached here, so we can detect or analyze this deletion. Now, if the translocations or other rearrangements are detected using probes of different colors, okay, if translocation or other rearrangements are occurred, can be detected by using the different types of the different colors of the probes, because these different probes are complementary to the regions on each chromosomes taking part in the translocation. It means that two chromosomes, sorry, these two probes are attached to the chromosomes. For example, this is one chromosome. For example, it is a chromosome one, and it is a chromosome two. And the yellow probe will be attached here, okay, and the green is attached with the chromosome two. So, if translocation is occurred between eight, okay, the parts are exchanged to the each other. The new derivative chromosomes in which the one part is of the chromosome 1 and the other part is the chromosome 2. And there is the two colors will be shown. There will be two colors will be shown here. One is green and the other color is shown will be here is green. Okay. And by this we can detect there is a translocation occurs in the chromosomes. Chromosome 1 and chromosome 2. Because in the normal chromosomes, the two colors are far apart, but in the translocation, the colors are joined. As here is also right, our translocated chromosome will combine two of the probe signals. Okay, it will the combine the two colors. As here, the two colors, one is the green, blue, and the other color is the green. But in the translocation, the two colors are joined, and these two colors will, when combined, it will produce the different types of the color. That is yellow, because they mix the color, and that mixed up color it produces a yellow color. And the second type of the probe is dual color break apart. For example, we use two color probes on the same chromosomes. Okay, two color probes on the same chromosome, and it is a normal. Okay, it is a normal chromosome. One probe is attached here, and one probe is attached here. If the translocation is occurred by breaking apart here, and this part is exchanged with the other chromosome. Okay, it's for example this. Okay, the part is exchanged between these two chromosomes, this chromosome and this chromosome. So by this, The probes will be break apart. Okay, as we see here, the two chromosomes, sorry, the two probes are attached, are combined. But in the translocation, the two probes are break apart. Okay, the third type of the probes is centromeric probes. Okay, it attaches with the centromere. For example, it is the centromere and it is the arm of the chromosome. Okay, then the probe, for example, the yellow probe is attached with the probe, with the centromere. The centromeric probes are designed to hybridize to the highly repetitive alpha satellite sequences surrounding the centromeres. Okay, in the centromere, the sequence present is called alpha satellite sequence. Okay, alpha satellite sequence that is very highly repetitive. Highly repetitive means if T is present in the alpha satellite, the every nucleotide is T. Okay, every nucleotide is every nucleotide is T. So it is very highly repetitive. Okay, and the probe is attached. The probe is designed to hybridize with the alpha satellite sequence that is present in the centromere. These probes detect the anosome of any chromosome. If there is any trisomy or the monosomy or the triploidy or aneuploidy, if occurs between the two chromosomes or between the chromosomes or in the chromosomes, that can be detected by the centromeric probes. The fourth type of the probes is telomeric probes. Okay, actually, for example, it is a chromosome. So, okay, and the ends of the chromosomes are called telomeres. Okay, this part is telomere, this part is telomere. The function of the telomere is actually uh, it prevents to the chromosome shortening. Okay, and the second thing is it also prevent the chromosome from the fusion to each other. Okay, now the telomeric probes are attached with the telomeric region. Okay, these are useful for the detection of structural abnormalities. Okay. The structural abnormalities such as cryptic height. Cryptic means height. The height translocations or the subtelomeric subdeletions that are not easily visualized by the standard karyotyping. And the probes are attached here. Okay. So it can detect the translocations or the subtelomeric deletions. Subtelomeric is that is the region is subtelomeric mm, that is beside the telomeres. So if there is any deletion in the subtelomere, okay, or there is a translocation that is a cryptic translocation that are hidden in the standard karyotyping, that can be detected by the telomeric probes. Now, at the end, talk about the metaphase fish. As we say, there are two types of the fish. One is the interface fish, and the second one is the metaphase fish. Actually, in interface fish, we don't have to grow the cells, but in the metaphase fish, we have to grow the cell. But application or the importance of the metaphase fish is we can analyze even the small region. Okay, we can actually analyze the small region that can not be visible by a regular chromosome bending. Okay, in the bending we analyze the whole chromosome by making a bands on it. Okay, for example, the 80 ridge region is uh, black, the this region is a light region. Okay, by this we can analyze the chromosome. Okay, by use the bands. But the small regions in which the deletion or the insertion of the application occurs can't be detected by the bending. Okay, this is the limitation of the bending. But by metaphase phase, because in the metaphase phase, um, the probes are attached to the complementary region. So by this, we can analyze the small regions. Okay, we can analyze the small regions that are not visible by regular chromosomes. The probes that cover the entire chromosomes. Okay, in the metaphase phase, the probes are not regionally attached or bind with the complementary region, but in the metaphase, the probes cover the entire chromosomes. Okay, it attaches with the entire chromosomes or the whole chromosome pairs. Okay, are valuable for the detecting these small or complex reagents. Okay, because the probes are attached to the whole chromosome, okay, or to the entire chromosome. So the small rearrangements or the small deletions or the small regions that are not visible by the regular chromosome bending can be detected by the metaphase fish. Okay, by mixing combinations of five floors and using special imaging software can distinguish all 23 chromosomes by chromosome specific colors, in which we use five floors means we use the probes with the five fluorescent dyes, okay, and special imaging software in which the different types of special filters are used, okay, and by this we can analyze or detect or distinguish the 23 chromosome pairs, okay, and by this we can analyze the whole genome, actually the whole genome of the cell of the any organism, and the metaphase fish has this importance. In the metaphase fish, the analysis can detect abnormalities that affect the multiple chromosomes. Okay, and if the problems are occurred in the multiple chromosomes, okay, and the abnormalities that affect the multiple chromosomes can be analyzed by the metaphase fish, sometimes found in the cancer cells. Okay, in the cancer cells, the problems are occurred or the effects or that affects the multiple chromosomes. Okay, and that can be analyzed by immortalized cells that are actually found in the cancer cells or the immortalized cell lines that can be detected and analyzed by metaphase fish. In the metaphase fish, the telomeric centromeric probes are also applied. Okay, and because it binds with the whole enzyme, whole chromosome, so telomeric probes that attach with the telomeres. Okay, the centromeric probes that attach with the centromere are also applied in the metaphase chromosomes. Okay, to detect aneuploidy, because by this can detect the aneuploidy and the other genomic mutations can be detected by metaphase fish. And the procedure is same like karyotyping, in which because in the metaphase stage we have to grow the cell, so we have to culture the cells for 72 hours and about 45 minutes before harvesting. Colsamid or colchicine is added to culture to arrest the dividing cells in the metaphase. Okay, so the metaphase arrest is also required. So colsamid is also added, and the cells are suspended in the hypotonic solution. Okay, like the KCl, potassium chloride, and the why we used hypotonic solution because the genetic material of the genome is present inside the nucleus or in the cell. Okay, so we have to remove this nuclear content out of the cell. Okay, the probes are not attached inside the mm, cell, but we have to remove this nuclear content out of the cell. Okay, so for this we have to use the hypotonic solution like the potassium chloride, and at the end we fixed it by using methanol or the acetic acid in the 3:1 ratio. And the fixed cell suspension is applied to an inclined slide and allowed to dry. And at the end, we inclined the slide and allowed to dry. And after the slide is actually the last slide in which we whole procedure we done. And at the end, we dry it, and after it, we analyze it by using fluorescent microscope. Procedure is same. A second treatment is 70% acetic acid. After this, we use the 70% acetic acid that actually improve the chromosome bending, spreading. Okay, the metaphase rest or the other things and metaphase spreading can be improved by using the 70% acetic acid, and it also decreases the background noise. And once the slide is dried, hybridization proceeds as for interface fish. And after this, we use the hybridization process as for the interface fish. The next procedure is same like the interface fish, in which we hybridize the probes with the genetic material. And the combination of different locus-specific probes, because we use the different locus-specific probes and chromosome paints, can be used simultaneously, yielding more information than carrying out separate experiments. Okay, because in the regional probes, okay, that attach with the points of the chromosomes, okay, that point that attach with the complementary sequence of the chromosome, okay, and by this we infer that the chromosome is actually deleted or amplified. Okay, but in the metaphase phase we use the combination. Okay, we use the combination of different colors that are attached with the same chromosome but at the different locus. Okay, so by this we can yield more information, uh, more information that are better than we use in the separate experiments in the multiple specimens. Okay, we collect the different samples, then we use the interface fish separately. In the metaphase fish, we can done in the same stroke. And the multicolor fish identifies specific chromosome regions based on presence or absence of the probe. Okay, we also use the multiple colors probes of the fish, by which if the probe is present, the information is somewhere else, and if the probe is absent, the uh, information is somewhere else. Okay, so we used it, and also we can use the special filters. Thank you so much for today. Hope so you understand the fish. It is very important technique, very important procedure in molecular diagnostics. Allah Hafiz. Goodbye.